Everyone. I think we are at 4:02, so I'm going to go ahead and get us started with a very, hopefully a quick introduction of our speaker today. Is it not on? Oh, we were just talking about this.
It is on. I think you just have to be right close to it.
Okay, there we go. All right. Okay, so it's my pleasure to be introducing Professor Emilie Snell Rood from the University of Minnesota. I first kind of encountered Emilie's work actually in graduate school whenever I was first thinking about the molecular basis of developmental plasticity. I was working in Drosophila at the time, and I was finding these papers actually about work that she was doing in horned beetles. But anyone if you've taken a look at her work, you recognize that it's expansive in terms of the ideas that she's thinking about, in terms of the elegance of the experiments, the theory that's covered, and there's just a lot of different ideas that Emilie is clearly thinking about, kind of the consummate naturalist, which has been kind of really amazing to see. And it's also reflected in the kind of diversity of people that were interested in meeting with her. Now our kind of point of convergence is in monarchs and thinking about behavioral kind of plasticity there. But what I'm really excited about and interested in is Emilie taking these foundational ideas in theory and now much more explicitly bringing them into applied context, thinking about conservation and other kind of applications, biomimetics, it ranges lots of different areas. So with that, please join me in welcoming Dr. Emilie Snell Rood and her presentation dealing with anthropogenic environments, why some species thrive, and how we make the best of marginal habitats.
Well, thank you so much, Andre. That was maybe one of the nicest introductions I've ever received. That's very, very nice. Thank you. Can everyone hear me okay? All right. So it's really a pleasure to be here. I've never been to the University of Michigan. It's wonderful. It's an honor to be invited. I've had a really wonderful day of talks, and I look forward to talking with more people tonight and tomorrow. So thank you so much for having me, and thank you, Andre, for inviting me and hosting me. So today I'm going to talk about 3 different sets of studies that fall under this general umbrella of dealing with anthropogenic environments. But first, I just want to acknowledge that this work is done in collaboration with folks in my lab and many, many collaborators who I will try to highlight throughout the whole talk. And these are just a couple of pictures of folks in my lab from over the last decade. So a lot of our work is motivated by anthropogenic environments. So the environments that animals and plants deal with today are drastically different from what they are used to over their evolutionary history, whether you're thinking about the spread of invasive species, the construction of new habitats like urban areas, the conversion of habitat to monoculture agriculture, or the introduction of novel pollutants. And so we're broadly interested in why some species thrive. So say this randomly chosen example of a cabbage white butterfly, which is found on every continent except for Antarctica. It does well with all kinds of stressors. And yet this other species, the West Virginia white, in the exact same genus, very closely related, that is incredibly specialized. It feeds on one genus of plants and is very restricted in its range. So how do we have this kind of variation, and who is thriving in the anthropogenic environments, and what does this mean for conservation? Because we're also very interested in how we can mitigate species declines. There are any number of depressing statistics that I could show you. This is one from a paper earlier this year focused on butterflies that just summarizes across the last several decades. Species in the US, the majority of them are in decline. So this is just a histogram showing the number of species in decline versus the ones that are increasing. And many of these are declining at rates of like 20 to 30% a year, which is horrifying. So what can we do from that as people who care about biodiversity, ecosystem function, all of this? How can we mitigate these species declines? There are many number of causal factors. Many of them boil down to habitat destruction, habitat loss, and disturbance of habitat not being as high quality as it used to be. And so then we think about, well, how can we add habitat? Are there places that we could preserve, that we could buy up and make wonderful, pristine land for organisms? Well, in the United States and most of the world, we're already using this habitat in various ways for human things. The land that is tied up in sort of pristine states like wilderness or national parks or state parks is a very small percentage of land use in the US. Much of it is already being used in agriculture as pasture land, timber, a lot in urban and residential use. So what can we do about this? So there have been many, many pushes to think about, well, how could we restore habitat on the edges or pockets of habitat? So for instance, just in urban or suburban areas, there are thoughts of, well, maybe we could make lawn into pollinator lawn. Or this fine gardening issue that's talking about how to create a beautiful pollinator haven in this pocket of an area in the middle of a city, maybe on a roof. So there have been more and more pushes to think about pockets of habitat, which is a great idea, but at the same time, there are risks associated with restoring those pockets of habitat. This is a pollinator garden and a rain garden that is receiving runoff from these roads. This is a community garden that supports a lot of pollinators. This was actually the first community garden I gardened in St. Paul, that's right on the edge of a massive railroad, so there's huge heavy metal input there. So there's this broader question of, in this push to create and restore habitat, are we basically setting up these ecological traps? So today I want to give a flavor of some of the work that goes on in my lab. I'm going to talk about 3 different things that illustrate the general questions that we ask. First, this question of why some populations or species thrive in these novel environments and others do not. And specifically, I want to talk about some recent work where we were thinking about responses to novel toxins and the role of evolutionary history with plant defensive chemicals. Then I want to talk about a lot of our work is thinking about these restoring pockets of habitat and whether we're creating ecological traps. We've done a lot of work in this context in roadside habitats and also more recently in urban areas. We've done a lot of work on heavy metals, but today I'm just going to give an example on some of our newer work on microplastics. And then finally, I want to talk about thinking about some implications for environmental design. And this is more speculative work that I've just started to think about in collaboration with some architects and people in the College of Design. All right, so we will start, and I will mention that a lot of this work is nested within the Minneapolis-St. Paul Urban Long-Term Ecological Research Program. So we have 2 LTERs at the University of Minnesota, one Cedar Creek, and then a relatively newer one where basically the Twin Cities is our field site. And we are studying the ecology of the Twin Cities. So I'm going to start by thinking about this question of why some populations and species thrive in novel environments and others do not. And specifically here, thinking about novel toxins. And this is work that is done within the MSP LTR, especially in collaboration with the postdoc Lindsay Kemmerling and an honors undergrad, Ashley Darst, who's now a grad student at Michigan State. So this work was focused on the question of novel toxins as a subset of novel human environments. So humans synthesize, I think, 70 or 80,000 chemicals in the US alone. Many of those are for as pesticides. We also are, there are many pollutants in waste as pollution. We have microplastics accumulating everywhere. We dump things like salt on the road. So organisms are confronted with many chemicals that are new or at levels that are new relative to their evolutionary history. And yet they vary in how they can cope with these novel toxins. And so we're interested in why species vary in their ability to tolerate these novel toxins. So this is a graph from a recent meta-analysis that looked as an example in variation in tolerance to a neonicotinoid pesticide. And the main thing I want you to see here is that across all of these different orders, there's a lot of variation in how tolerant they are across the orders and within the orders. And so we want to know why that is. And then this probably has consequences for thinking about adaptation to these novel toxins. So for instance, species like this moth that has evolved the ability to completely resist certain pesticides through adaptations in various enzymes and receptors. The fact that they could evolve that in the first place meant that they could tolerate it at least to some degree in the first place. So why do species vary in their ability to tolerate novel toxins? We were interested in 2 hypotheses. And to test this, we were focused on heavy metal pollution as a novel pollutant. Lead, in particular, is not a novel chemical. It's a natural element. But the levels that we see it in today's ecosystems, you might consider novel. Humans basically pull it up through mining, put it in various products, and that redistributes it across the landscape. Lead also is also important for thinking about human health. So the CDC says that no level of lead exposure in humans is safe. It has major effects on cognitive development, and of course, large amounts of lead can result in poisoning and death. So lead can be toxic to a lot of organisms, not just humans, and the levels that you see it in association with humans can be at novel levels with respect to evolutionary history. So we were interested in two hypotheses using lead as an example. The first one was a hypothesis that we borrowed from the pesticide literature, and this is the idea that evolutionary history with plant toxins might predispose some species to deal with novel toxins. So this is an idea that's been around and suggested by May Berenbaum, that species that have evolved pesticide resistance might be those that can generally deal with toxins that plants usually produce as a chemical defense, and they just co-opted those mechanisms to deal with novel toxins. More specifically, we had a more specific version of this hypothesis with respect to heavy metals, because heavy metals cause a lot of oxidative stress. So they like to grab electrons and cause all kinds of oxidative stress, free radicals, DNA damage based on that. And because of that, we were specifically interested in plant defensive chemicals that also happen to be mutagenic, because those are also causing oxidative stress, free radicals, DNA damage. And so we thought there might be overlapping underlying mechanisms such that if some species could deal with a plant toxin, like this is a pyrazidine alkaloid that also happens to be mutagenic, then maybe they could also deal with high levels of heavy metal exposure that also causes free radicals and DNA damage. So that was our first hypothesis. As we started to dig more into the literature, we also decided to test a second hypothesis. There's a general idea that range size is related to dealing with environmental stress. It's mostly been tested with respect to temperature stress, but there's an emerging idea that it might be related to stress in general. So we also were testing whether species with larger ranges could also cope with novel toxins, presumably because if you have a larger range, greater environmental variation. This is selected for greater degree of plasticity and dealing with that environmental variation, which might have a positive feedback loop over evolutionary time. So here we're also contrasting species that vary in range size and asking whether this one in general might be more tolerant than, say, this one here. So how do we actually test this? So we were focused on butterflies as a system. Butterflies are diverse, but they also vary quite a lot in what they feed on as caterpillars. And generally, species are fairly specific to one or two plant families. Their phylogenetic relationships are fairly well worked out, maybe not as well as some groups, but relative to a lot of insects, we have a pretty good understanding of how they're related. And because people are obsessed with butterflies, we have a pretty good idea of their natural history and what they feed on and when those host shifts happened. Because different species are specialized, so say this is all within the family Pieridae, these guys feed on mustards, these guys feed on pine, these feed on asteraceae, these feed on legumes, these feed on blueberries, we can quantify sort of what their evolutionary history has been with different plant groups for a certain amount of time. These plant groups also differ in what defensive chemicals they have, and we struggled for a long time in trying to figure out, okay, we want to actually somehow have some measure to quantify the nastiness of these plant defensive chemicals across all of these plant groups. So what we ended up doing is using a standard assay for mutagenicity called the Ames test. So we wanted to basically say, across all plants, what is the probability that some plant group that a butterfly feeds on as a caterpillar is mutagenic? So to do that, we amassed as many different studies as we could find that use this mutagenicity screen called an Ames test. The basic way it works is you take a plate of bacteria and you apply some potential mutagen. In this case, it would be an extract from a plant. The bacteria cannot metabolize something in the plate, but then as they mutate and they can acquire the ability to metabolize that thing in a plate, they change color. And so this screen has been used since the 70s. There are a bunch of studies across different plant extracts because it's a fairly standard screen. And so we amassed as many as we could and then asked, okay, how does this vary with respect to plant family? So this is just a summary of what we found across plants. I don't expect you to read all of these. These are all just different plant families in order by by mutagenicity. So this was, we have a very long extended methods section for this paper that talks about how we did this. I'm happy to talk more about that. But basically, we end up with some plants that are relatively less likely to be mutagenic relative to other plant families. And just to kind of orient you, the plant family Aristolochiaceae, which is a known mutagen, a known plant family that causes kidney cancer in humans, is way up here as the third most likely to be mutagenic. So we take this data and we align that with the host use data of the butterflies to get an idea of what is the chance that they're co-evolving with mutagenic chemicals. All right, so that's the dependent variable. So then the independent variable. The response variable is their tolerance to this novel mutagen, this lead levels. So here what we did was we basically went out to collect butterflies across a gradient of lead exposure in the Twin Cities. Ideally, we would want to take like 20 or 30 species of butterflies and actually raise them in the lab and figure out at what levels they're tolerant to, but that's really hard to do, especially if they feed on different plants. So we did the next best thing by going out and collecting them in areas that we assumed would have higher rates of pollution than others. We chose our field sites based on maps of childhood blood lead levels, which in Minnesota, it's standard to screen all children at age 1 and for lead, and that is public data. You can see, this is by census tract, you can see areas of elevated blood lead. We've done a lot of work thinking about what is causing this variation, which is, I'm also happy to discuss those things. We've also done a lot of work in these two census districts in East St. Paul, which have the highest rates of elevated blood lead in the state. So we chose our field sites for butterflies along this gradient. This is a map that's somewhat expanded, so you can also see our control sites. Here you can see where the butterfly collection sites are. Basically, any green space where we could collect butterflies is where we collected butterflies. So we went out, collected them along this gradient. Interestingly, and then we measure load of lead in their thorax. Interestingly, butterflies that are growing up in the same areas where humans have elevated blood lead also have elevated tissue. So this is the human blood lead levels, the census districts with high or moderate lead, and butterflies in those areas also have elevated lead. We see the same patterns in bees, which was shown in the dissertation of this grad student, Lauren. We have done some work on the routes of exposure in the butterflies, so I'll mention that briefly. Interestingly, it seems like the butterfly exposure is primarily through direct contact with the soil, in addition to air pollution, rather than through the plant. And something similar is probably going on with the humans as well. Things are probably similar with the bees as well. So in some of Lauren's work, she found that bee species that nest below ground and are probably having greater contact with the soil have elevated levels of metals in their tissue, in this case, copper as well as lead. So lead exposure in the butterflies, we took a whole bunch of measures on their brains, fecundity, body size, just to look at performance effects. Lead exposure is stressful, but not nearly as much as we thought it would be. So this just shows across all of the species we looked at, as butterfly lead content goes up, their body size goes down, but only slightly. It is significant, but only slightly. We think one possible reason why is because overall lead levels in the Twin Cities and indeed in the US are much lower now than they were 100 years ago. So this is, as an aside, we did a study using the mammal collection at Minnesota and just went back about 100 years looking at punches of skin of mammals, of 4 different species of mammals. And what you can see is that overall, this is on a log scale, levels of lead in the tissue declined by about 5 times over that time period. So overall, lead is stressful, but not nearly as much as it probably was 100 years ago. So back to the comparative aspect of this. So across all of these butterflies, we looked at how the lead burden was associated with species. So some species, say like cabbage whites, could tolerate much higher on average levels of lead than other species like this grass-feeding satyr. So this is just showing species variation in the butterfly. And so then we finally put all of this together in the comparative study. And so here's the punchline, is that both evolutionary history and range size are important predictors of lead load. But the range size was actually much more important. We looked at some other metals as well. So this just shows, so in these graphs, to orient you, each point is a species. So for instance, cabbage whites are here, monarchs are here. This shows the average mutagenicity of their diet based on what they feed on and the probability that that plant family is mutagenic. And then this shows, in this case, the maximum levels of lead we saw in these butterflies. So there's a positive relationship, and this p-value corrects for phylogenetic relationships. And there's even a stronger relationship with respect to range size. If you look at other metals like arsenic and cadmium or manganese, you see the same effect with respect to range size, but not host plant mutagenicity. I'll just end this section by noting that while this was a field study, we do have some data from lab rearing experiments that provide some consistency. So this was work done by a grad student, Alex Shepard, where he reared monarchs and cabbage whites on different increasing amounts of zinc. And what you can see is that the monarch, as zinc goes up in their diet, start to have significant effects on survival around 350 or so ppm. Whereas for cabbage whites, they actually have an increase in survival. So they're probably activating stress responses that are having beneficial effects, at least at these levels of zinc. All right, so what do we make from this section? So we find support for this idea that evolutionary history with mutagenic chemicals can explain at least some variation in the ability to tolerate elevated lead along an urban gradient, but that variation in range size is a much stronger predictor. And so this suggests there's probably a role of selection on plasticity and stress tolerance in general that I think should be dug into in more detail with respect to the mechanism. Existing lab studies show similar findings, but we do need more controlled measures of tolerance to complement field studies across a broader range of species. All right, so next I want to transition into thinking about, okay, well, we want to restore pockets of habitat. Many of these pockets of habitat are polluted. Can we do that, or are we creating some kind of ecological trap? And I'm specifically going to talk about roadsides as an example. We've done a lot of work on roadsides because in the last couple of decades, there's been a huge push to restore roadsides for pollinators, even at the level of departments of transportation prioritizing pollinator restoration in their funding. So why roads? So roads we have a lot of roads. We actually have more linear mileage of roads in the United States than we have streams and rivers, which is kind of depressing. But it also means when you think about the rights of way along those roadsides, that in some ways roadside habitat is functionally equivalent to riparian habitat. And that's a massive amount of potential habitat if you're thinking about restoration for something like pollinators. Roadsides also can be very good habitat for pollinators. So roadsides, at least in Minnesota, have a ton of milkweed. They can support per mile over 2,000 stems of milkweed, the host plant of monarchs. They can also host a lot of other pollinators. So in roadside surveys that we did, we found quite a lot of butterflies that are pretty common along roadsides, including monarchs, which were the 6 most common species that we found. We also see a lot of bumblebees there, including about 5% of roadsides in Minnesota host the endangered rusty patch bumblebees. So there's a lot of potential there, but there's also a lot of concern that maybe they're ecological traps because roadsides also have pollutants. There's still residual lead from the days of leaded gasoline on many roadsides. There's also continual shedding of heavy metals from cars, especially brake pads and general car wear and tear. There's also pesticide spillover from adjacent agricultural fields. So there's this concern about contaminants. At the same time, roadsides might even lure pollinators in because they have elevated nitrogen from exhaust, which has been hypothesized to cause insect outbreaks along some roadsides because the nitrogen content of the leaves in those plants can sometimes be elevated. Similarly, dumping salt along roadsides can elevate sodium content along roadsides, which can attract insects. So are we just drawing insects in and then killing them based on the toxins that are there? So we've done a lot of previous work on salts and heavy metals. I'll mention that briefly at the end. But today I want to talk about some of our unpublished work on microplastics. And this is work done with a number of undergrads and one grad student in the lab. So microplastics are a contaminant of emerging concern, in part because we're finding that they are everywhere we look, including in our blood, but we don't quite understand how much we should be freaked out about them. So basically, plastic just degrades into smaller and smaller pieces, and plastic is in pretty much everything we build, including the chairs and the carpet and our clothes and everything. Roadsides also play a role in what you might call the plastic cycle. So a lot of times we're thinking about microplastics in water because as we wash our clothes, you get fibers in the water, it accumulates in water. But a lot of the terrestrial microplastic pollution actually comes from tire wear and tear. So about almost 30% of microplastics in terrestrial pollution come from tires. So for tires, shedding synthetic rubber. So you're thinking about runoff, and also from paints and other things on the asphalt are also made of plastic. So those things are wearing down and running off into the roadsides or getting into road dust and blowing up to a mile from the road. So we were interested in what this might mean for roadside habitat. And the first thing that we did was go out and just ask, okay, for plants that are growing along a roadside, what are we seeing in terms of microplastics on those plants? So Thea went out and collected plants along several high traffic and low traffic roads. This is just a shot of one of the high traffic roads in St. Paul. She collected leaves from a couple of species that are used by pollinators, and then we just asked what we were specifically focused on the tire particles. How many are accumulating on the leaves, and then should we worry about that? So we tried a number of extraction methods for these microplastics, and what turned out to work well was actually just looking at them under the microscope, because it turns out the tire particles look pretty distinctive relative to a lot of other particles. So these are just some images from a couple from milkweed and a goldenrod, and you can kind of see these little particles here. These are zoomed in. These are in microns here, so this one is about 300 microns wide. This is like maybe 20 microns wide. And we used this technique called FTIR to confirm that yes, they are the polymer that you see in synthetic rubber, which is styrene butadiene rubber. I should say the chemists that we worked on really did that part. We just gave them little tire bits. And 100% of the particles that we pulled off were this microplastic. We see significant differences with respect to roadside and plant type. So we'll start here with the large particles. So these are particles about 45 microns to about 300 or 400 microns in width. This is milkweed, this is a mustard, and then this is a goldenrod. And what you can see is that the plant leaves from higher traffic roads have more particles on their leaves than the low traffic roads. But for small particles, these are like 10 to 44 microns. There's no difference. And this is consistent with the idea that heavier particles fall out of the air faster than these smaller particles, which are getting blown around more. The next question is, okay, there are microplastics in these plants. Is this something we should freak out about? Or is this something that the animals living in the space can deal with? So we did some calculations to figure out, okay, what does this highest amount in, say, a milkweed translate to in terms of the diet? If you're a caterpillar munching on this leaf, in terms of what you're actually eating bite per bite, this should correspond to about 3.6% dry mass of the leaf. So we use that to make diets to ask how it affects the butterflies. So we weren't sure how this was going to affect the butterflies, and we actually thought, maybe we should start with a simpler polymer because tires have a lot of stuff in them. So we actually decided to start with ground up polyethylene, which is the polymer that you get in like your standard plastic water bottle that I was drinking out of at lunch. So we focused first on cabbage white butterflies because they're really easy to raise on an artificial diet, and people rarely get upset if you're feeding a pest butterfly plastic. So we raised them on diets from 0 up to 8% dry weight of the diet, and there was no effect on anything we measured. So this is development time. This is number of mature eggs. We also took body size measurements, and so there were no effects on these performance measures. We're like, okay, they're fine with this. Let's see if we can stress them out a little bit more. And as I hinted at already, the tires and these tire particles have a lot of other stuff in them. So we wanted to know whether, if we just fed them ground up tire bits, whether they would be okay with that. So this took a fair bit of troubleshooting, first trying to get tire bits that were small enough to replicate what we were seeing on the leaves. And what we ended up doing, we tried like grinding them with liquid nitrogen and a coffee grinder and all this stuff. What we ended up doing was buying from Etsy these like finely ground crumb rubber, because people apparently like to glue this to their shoes for some reason, and sifting it into the particle size that matched what we saw on the plants, and then putting it into the diets. So our diets ended up going from 0 up to, we decided to stress them out even more, up to 16%. And you can see the diets, like, they look black. They've got so much of this tire dust in them. Turns out the cabbage white butterflies are tolerant up to 8% in their diet. So this is development time here. You can see they start to slow down, but there's not a significant effect until you get to the 16% dry mass of the diet. But even then, look at the scale here. You're basically slowing them down by 1 day. They're still growing, which is kind of shocking to me, but they're like, oh, just slow me down by 1 day. There's no effect on overall survival, no significant effect. So we thought, well, maybe we know that monarchs are more sensitive than cabbage whites. Maybe monarchs would get stressed out. So we tried with an artificial diet for monarchs. Here we just went up to 8%. We have not analyzed all of the data yet, but at least the initial survival to late 4th instar, there is no survival effect. We've also taken data on lifespan. I just got an email from the undergrad. They just finished doing dissections yesterday for fecundity. So we'll have data on all of these other measures soon. But the take home here is that roadsides are polluted. Yes, not surprisingly, but the levels of microplastics do seem to be below thresholds of concern for caterpillars, at least with respect to survival and development time. And here I'll note that across our other studies, we see similar patterns for other pollutants like metals and salts, and other marginal habitats like urban green spaces. And just to kind of drive that point home, this is a graph that summarizes across a bunch of different studies on several different species of moths and butterflies. This is the level of minimum effect on either survival or body size for a bunch of metals, so cadmium, copper, nickel, lead, and zinc. So you can see cadmium is way more toxic than lead, which is way more toxic than zinc. And then the little stars here show the maximum level that we see in plants along roadsides in Minnesota. So in general, yes, these metals can be toxic to them, but the levels that we see are generally in the field are way below what would stress them out. So what does this mean with respect to this question of roadside habitat and whether we're creating an ecological trap? So in general, it's good news, but I just presented this to a bunch of transportation people a couple weeks ago, and so I added this. It's also not a license to pollute. It's not like, okay, let's just, yay, tire particles everywhere. Because we also know that these chemicals can be bad and in large amounts. And we also know that tires leach chemicals of concern into water, and that smaller particles are also more worrisome than some of these larger particles, so like nanoplastics. So the take home here is that we can proceed with thinking about roadside habitat for pollinators, but we should also continue interventions to reduce pollution. And to that end, I'll briefly mention some of the work that we're working on with respect to mitigation. We're doing a lot of stuff with biochar. So biochar is basically a fancy charcoal that you make out of biological waste. It's most commonly made out of timber or agricultural waste. You heat it at high temperatures under low oxygen, and you turn it into a charcoal that has these very small pore spaces. And those pore spaces give it a lot of interesting properties, including binding pollutants. Although the extent to which they bind pollutants varies quite a lot with soil chemistry. So we've been doing a lot of work testing biochar soil amendments in mitigating both metals and microplastics. There's been a lot of work on biochar and how it might reduce the bioavailability of metals in soils to plants, but only one study so far with respect to microplastics. So we have one experiment that we're just starting to analyze the data on, where we amended soil in community gardens with biochar and looked at microplastic bioavailability. And then we have a number of studies at polluted sites, at either current or past Superfund sites that have heavy metal pollution, and looking at biochar addition either to the soil or on the soil surface. So these are biodegradable biochar-based plasters that we used both for weed control and for soil improvement at the start of a pollinator planting. So I'm happy to talk about any of that in more detail as well, but that is ongoing work. All right. So the last thing I want to talk about is thinking about some weird or creative ideas. Yeah, creative is probably a better word than weird. Speculative ideas about thinking about design and intersections between sort of EEB type fields and design fields when we're thinking about urban environments. And a lot of these thoughts are things that came out of collaborations that I've had over the last decade with people in the College of Design, especially in architecture. I taught with several people, especially this wonderful guy, Billy Weber, a class on bio-inspired design, and started to think about some of these ideas. But I've also done a lot of collaborative work with this woman, Mary Guzowski, who does a lot of work on biophilic design. Malini does a lot of work on these biochar-based materials. And then Prasad we're collaborating to build sort of groups that do bio-inspired design. So a lot of this work has, I would say with the design lens, has come from working with them and thinking about, if we're thinking about ecological restoration, and if we're thinking about cities as ecosystems in general, those are areas where we intersect with design and design questions. And so especially as we've been thinking about the urban long-term ecological research, how do we think about designing a city? We usually think about designing a city for people, but the city is also harboring plants and animals and microbes, and many of those things are intersecting with the benefits or burdens of the city to the people. So we need to think about not only the human well-being side in urban design, but how those design goals intersect with ecosystem services like pollution filtration, stormwater capture, biodiversity, buffering climate stress, pollution, pollination, et cetera. So we've been trying to start to think about, well, how could we design a human environment to promote things that we care about as biologists? And there's been a lot of discussion in the design world with respect to ecosystem services, but I think we should broaden this question more, and I think us as biologists have a lot to bring to this design question. And so I want to challenge biologists to think about not just the ecosystem services part of it, which is what an engineer is often thinking about or a designer is often thinking about, but broaden this question and think about how can we design a human environment that promotes the development and evolution of resilient populations? And how does that intersect then with human well-being? So I realize this may be vague, so I want to give two examples, one with respect to development and one with respect to evolution. So this first one is a project thinking about how vegetative diversity and complexity in general promote cognitive development. And this is an idea that has come out of the literature, that has come out of the lab literature, but in superimposing that on thinking about how we currently design our environments. Because most human environments are incredibly simplified and predictable relative to how the environment usually is. We have monocultures in agriculture in our front yards. And if you are a grass-feeding butterfly, this is a very simple and almost easy environment relative to what you experienced in your evolutionary history. This is interesting because there's decades of lab work that suggests that growing up in a complicated or enriched environment in lab situations stimulates neurogenesis and stimulates brain and cognitive development. This is true in primates, it's true in rodents, it's true in crickets, it's true in fish, pretty much everywhere you look. So what does that mean? What does this mean then if all of the urban environments that we have are highly simplified and predictable? So to give an example of this, I want to talk about some of the work that we've done with butterfly host searching behavior, and just to illustrate what I mean by it. So here's some cabbage white butterflies in a very simple environment. This is a field that's just packed with their host plant. They lay their eggs on mustards, and every single plant that you can see here is a host plant for them, except for that grass. So as a female flies around, she lands on the plants, she tastes them with her feet, and if they have the chemicals associated with being a mustard, she lays an egg, which you saw right there. It takes her about 2 seconds to lay an egg. Off she goes to lay the next egg. This is a pretty simple, straightforward host finding environment. So as I mentioned, the caterpillars can only survive on plants in the mustard family. They pay attention to these glucosinolate chemicals, which is what makes mustard taste spicy to us, to identify the plants by touching them and tasting them. Now let's imagine a slightly more complicated environment. So here, the circles show the host plant, and everything else is a non-host plant. So if a female cabbage white, in this case, lays an egg on one of these other plants, which were geraniums in this case, the caterpillars would die. Those represent mistakes. So what happens in this case? So in this case, the females learn. So a naive female that's never seen a host plant will start by landing on lots of different plants and tasting them, and then over time, she selectively learns colors and shapes associated with what host is common in that environment. So here's a naive female. Sorry, this video is pretty bad, but here she's landing on a bunch of plants. She initially makes mistakes. She laid an egg real quick on a cabbage there, then she landed on a geranium, tasted that, lays quickly on a cabbage. Then a couple minutes later comes back, lays on a cabbage, and then she keeps there. She sampled a non-host, but she keeps going, sampled another non-host, keeps going, makes another mistake, keeps going. So you can, based on what they're landing on, you can tell what they're choosing. You can look at their mistakes and you can plot a learning curve, and their mistake rate drops precipitously. Over about a 30-minute period, they learn to selectively land on the host plant, and you can look at what mistakes they're making to see what they're searching for. So for instance, in this environment where it's a red cabbage, they make more mistakes on the red geranium here after they've learned it. Here they make more mistakes on the green one. So they learn color, they learn shape, they learn a lot of things associated with finding the host plants faster. Interestingly, you can then take this another, just want to make sure that's not for me, okay. You can take that another step and go from this environment, which has 2 non-hosts, to an even more complex environment that has 10 non-hosts that vary in color. So when this happens, you're making them learn something even harder. And it turns out that this degree of enrichment during that learning process stimulates brain development even more in these butterflies. So this was some work that I did actually during my dissertation, where I was then looking at the size of different brain regions. And butterflies that had just a day of experience host searching in this complex non-host environment, this one here, that induced the growth of larger relative volume of their visual, of the medulla, which is the visual part of their brain. So that's just one example of many that show that enriched environments matter for cognitive development. And I think this is important for thinking about urban design, because if you have green space, it basically suggests all green space in an urban environment is not created equal with respect to cognitive development. This type of environment would basically favor a butterfly that doesn't have to do much work, doesn't have to invest much cognitively, whereas this environment would favor the development of more cognition, larger brain in a butterfly, and that has further implications for surviving in a novel environment. So there's almost a positive feedback. So the last example I'll talk about here is thinking about the evolution side of this. So can we promote an evolutionary response to cities? And here I'll say, assuming that we should. There might be some cases where you might not want things to adapt to a city, but let's assume we want to promote biodiversity and have things deal with a city and adapt to a city. So here I want to briefly review the idea of evolutionary rescue. So this is from a literature where you're thinking about there's some environmental stress that causes population decline, and then at some point in time, that population evolves some adaptation that allows them to deal with that environmental stress, and so they're rescued by evolution. So this is the idea, but applied to a city where presumably everything kind of went extinct. And so it's more like the ability to move into the city, get a foothold, and then start adapting to that city. So how could we promote that kind of evolutionary response to a city? So the first thing I'll point out here is that that first step, niche overlap, matters a lot in the ability to get a foothold in the city in the first place. So a city to us looks like drastically novel and stressful relative to the environment a primate lived in. But we have a number of examples of species that thrive in cities, presumably because there's some degree of niche overlap. So it's been suggested that both rock doves and peregrine falcons do well in cities because ancestrally they've done well on cliff faces. And that's kind of similar to cities. So this graph, it's not very pretty. I wish I had a graphic designer to work with on this. But this graph shows thinking about, so two axes of the environment. And just hypothetically, a city is in this space, which represents somewhat novel conditions relative to historical conditions for species 1 and species 2. Species 1 at least has some degree of niche overlap with the city, and presumably that can give them some foothold in the city that provides an avenue for adaptation to that environment. So this kind of tries to quantify that. The degree of niche overlap between a species and a novel environment is going to be related to population size in that space. That's going to be related to effective population size, genetic variation, and potential adaptation to that environment. But then, of course, selection intensity is also going to decline. So as you're less stressed and as you have more niche overlap, selection intensity will be less. So there's probably some sweet spot of thinking about rate of adaptation to a novel environment where you have some foothold, but you also have enough of a population that you're adapting. So what does this mean with respect to urban design? So it means that the way we talked about it in this book chapter is thinking about some kind of pockets of habitat that allow footholds, that allow populations as sort of stepping stones into that urban environment. So things like a rain garden or a stormwater pond or a roadside planting or a garden or a green roof. These are pockets of habitat that animals might be able to move into. Then there's further selection in that space. In terms of, but you also have to think about the landscape context if you're thinking about selection. So of course, connectivity and corridors between the maybe rural areas or what you're thinking of as ancestral environment and the core of your novel environment, so the city. In this case, you have fairly low habitat connectivity, whereas here you have corridors and higher habitat connectivity. This means you would have higher gene flow, which means you would have less genetic divergence, but because you have more back and forth, higher selection on something like plasticity, which might result in a more resilient population in the end. So the point of this is I think evolutionary biologists actually have more to contribute with respect to thinking about urban design than is currently appreciated. And if you're thinking about, so this is a diagram of the Twin Cities, thinking about where the green space is, an evolutionary biologist approaching this might think about, well, we need more corridors here or here, but we also need to think about what are the other biomes, what are the other habitats that organisms are going to be coming from that would move into this space. And the Twin Cities sits right smack in the middle of 3 different biomes. So it's not just green space, it's also what is in that green space with respect to those ancestral environments. So some implications here that I'll end on. I think we can benefit from incorporating more behavioral ecology and evolutionary biology into thinking about the design of human environments. This is something that's been discussed a lot in the design field. So this is a paper I really like in a journal called Cities that talks about sort of principles from biology and ecology for the design of cities to promote biodiversity. So people in design are thinking about this. They are talking about biology a lot, but often without biologists. And so I think there's more to be done by interacting with those groups. So can we build habitats in urban and suburban areas that promote the development of more flexible and resilient organisms? And can we model how the type and structure of green spaces might affect gene flow and selection on things like plasticity that could affect survival and resilience of populations? So I'll end by just reviewing and noting that this kind of, I hope I could just give a flavor of some of the stuff that we do in the lab, thinking about variation in species in response to human environments, what that means for conservation, whether we can restore pockets of habitat, which I think in general we can. The benefits outweigh the risks, even though there are some risks of restoring these pockets of habitat. And that I think we have a lot to do in terms of there's a lot to benefit in terms of incorporating biologists with urban design and people outside of biology that are implementing things relevant to design, or to biology, sorry. All right, so I will end by acknowledging the various funding sources for this work, and thank you again for the invitation and for listening.
Any questions? All right.
Yay, thank you. You really do have to put your face in this. I had a question about your second vignette with the microplastics from the tires. And you showed that the caterpillars were pretty darn resilient to even high concentrations of the microplastics. But you showed them on leaves of their host plants. And we know that when a caterpillar eats its host plant, it's encountering like all kinds of physical and chemical defenses from the leaves that can sometimes interact. And so what I'm wondering is whether you thought about or plan to do any assays where you feed the caterpillars the leaves with and without the plastics on them.
With the plastic on the leaves?
Like with and without them on them, to see if it's the combination of ingesting the plant defenses and the plastics that could potentially spell trouble.
Yeah, so in the experiment, we were using an artificial diet. So we only added the plastics. So you're saying, but I could see if they're on the leaves and they're inducing defenses to the plant chemicals, then you're thinking they might be even more resistant to the stress? Is that the idea?
No, I'm thinking that if they are having to deal simultaneously with plant defenses and microplastics,
so that would stress them out.
That maybe it's that interaction that could end up making the microplastics a problem that you wouldn't see when they're on their own.
Yeah, that's an interesting point. So I will note the artificial diet does have host plant material in it. Cabbage whites are really good at dealing with the defensive chemicals, and I'd want to think about maybe there are other ways we could stress them out and see whether multiple stressors could have that effect. So yeah, that's a good idea.
Close do I have to get to this? Really close to it? Okay. So for the second one and the first one, you used monarch butterflies, and they're pretty well known for using their host plants as a form of self-medication. So I was wondering if you were planning on looking into whether those toxins and the microplastics might have some kind of secondary effect on their health, interacting with something like parasites or disease.
The microplastics might interact with parasites like as a second stressor, kind of?
Like whether that might have an indirect effect on parasite resistance in some kind of way?
They're packed full of plastic.
Yeah.
What you're saying?
Yeah, whether that might.
Yeah, so we are interested. There's a student currently in the lab who's using paper wasps to see whether they can discriminate against caterpillars that are loaded with pollutants. She's currently working on metals more than the plastics. I think interestingly, the paper wasps usually don't eat the guts of the caterpillars, and most of the microplastics I think are just passing straight through their gut. So I think if they avoided the gut, they would avoid most of the toxins in that. But if you're a bird, then yeah, you would be ingesting the whatever's in the gut as well. So I think it would depend on the predator. I don't know if that answered your question or if I just went on a tangent about paper wasps, but yeah, thanks.
I was wondering, you were talking about a bunch of different toxins, and if any of them have any bioaccumulative properties, and if like the longevity of an insect, like I know in monarchs certain ones, migratory ones live longer, if that kind of effect is anything that if you know anything about that, or if there's any studies that you're familiar with.
That's a great question. So the question was about bioaccumulation and how it might vary with the toxins. It varies a lot depending on the toxin. So, and even within the toxin. So like there's debate in the literature about how lead bioaccumulates, and our mammal study suggests that at least with these, and it probably depends on the form of the lead. that it's bound in chemically. At the mammals we worked on, it suggests it does bioaccumulate because it was higher in the higher trophic level species. For the microplastics, I would guess, so there is some work on aquatic insects suggesting that some of the smaller micro and nanoplastics probably are bioaccumulating. For these, I think they're big enough that they're probably passing through, so they're not bioaccumulating. But yeah, it depends on the toxin.
Sorry. I was wondering if you see any interest-specific variation in color or pattern among your butterfly populations in areas that are with heavy metal concentrations that are high versus low.
That is a great question. So we did a study on that, and we haven't analyzed the results, so which I'm embarrassed to admit because it was actually a while ago. But there's a grad student who now wants to return to that data set. So we were specifically interested in melanin upregulation and dealing with oxidative stress, and so and interactions with immunity, because insects use melanin in their immune pathways. So we raised things on high and low metal diets and looked at both wing coloration and melanization and antioxidant effects. And I don't know what we found, but hopefully we'll know that soon. I will say there was one study we did on cabbage whites where we were looking at, where we were feeding them novel plant toxins and looking at resistance to novel toxins, and that did show a trade-off with melanization in the wings. And I can send you that paper if it's from 2020.
Here you go.
Really cool work. Thank you so much. I had a question that kind of relates your last vignette to your second one, because you saw that there weren't impacts of the tire rubber on the butterfly performance, and you saw that like the levels of the contaminants are far below like the minimum effect threshold for a lot of butterfly species, but that's looking at like lifespan, right, and like time to development and survival, but you talk about cognitive development. So could these toxins be having like impacts on the cognitive development of the butterflies that are having effects that you might not see in a lifespan or survival curve? Yeah,
that's a great question. So we did look at brain development with some of our heavy metal experiments and saw no effect. So a lot of heavy metals are also neurotoxins, and so we were actually kind of surprised that we even did lead manipulations that had no effect on the butterfly learning. So I don't know how they're so resistant to that in terms of neurodevelopment. We have not done it with respect to microplastics yet, but that's a great question. Yeah.
Thank you.
Okay, in the same vein or similar vein, you've done a lot of great work talking about pollutants or other kind of anthropogenic, I don't know, human things that potentially disturb and mess up other things. And so something I've been thinking about specifically with monarchs, and as you mentioned, here in Michigan too, there's just like fields of milkweed that love the roadside. But something I've thought about, and maybe it's my own lack in knowledge of milkweed larval development as it relates to migratory success, light pollution. So we know that, if I go catch some fall monarchs and I even keep them in my home for like a night with artificial light, they just like fail to migrate or fail to show behaviors associated with migration. So do you have thoughts about like headlights or street lights?
Yeah, I do have thoughts. And and actually there's a proposal, there's a potential postdoc who might come to the lab to work on that and ask whether light pollution affects diapause induction, because I would guess based on everything we know that it does. Either ambient levels or localized levels. I don't know about headlights, but like a house porch light or something like that, and all the rearing stuff in monarchs suggests that it probably does. But I'm also interested in what kind of interventions could work, because I don't think you're going to convince the city to turn off their lights. So could something like a shade in your yard mitigate that effect at night? I don't know. But yeah, it's a great question.
Yeah.
All right, everyone. please join me in thanking Emilie again for that wonderful talk.
Thank you for having me.
Well, thank you. It's such a... I kind of hate this thing. And I don't know if it's... I don't know how to turn it off, quite frankly. So, let's see. Sorry. I think it does live here. And I kind of wish...